As Griffith could not isolate or identify the transforming principle, so many scientists repeated his work, and after about 15 years, Avery, MacLeod, and McCarthy succeeded in isolating and purifying the transforming substance in 1944. They showed that it had a chemical composition closely matching that of DNA and quite different from that of protein. So it provided a clue that DNA is the genetic material. They showed that proteolytic enzymes are those enzymes which break down proteins, and they include pepsin, trypsin, trypsinogen, and they showed that these enzymes had no effect on the transforming substance. Secondly, they also tested ribonucleases. Ribonucleases are also enzymes which break down RNA. So ribonucleases also had no effect on that substance. Thirdly, they showed that the enzymes capable of degrading DNA, they destroyed that transforming substance. Let us see the experiments conducted by Avery, MacLeod, and McCarthy. So, in their experiment, they took heat-killed S-type bacteria and they cultured these bacteria. After culturing, they homogenized the culture and filtered it. These filtrate was treated differently. For example, they took the filtrate and they treated it with RNAs to destroy all the RNA present in the filtrate. They also treated the filtrate with proteases, and the purpose was to destroy all the proteins present in the filtrate. And in their third experiment, they treated the filtrate with DNAs so that any DNA present in the filtrate may be destroyed. After treating the filtrate with different enzymes, they put that filtrate into R-type bacteria, into the culture of R-type bacteria. So all these were added into cultured R-type bacteria. So after culturing some days, they found in the first experiment both the S-type and R-type strains were present. In the second experiment, in which the filtrate was treated with proteases, both the S-type and R-type bacteria were present. In the third experiment, in which the filtrate was treated with DNAs, no S-type bacteria were present. In the above two experiments, the S-type bacteria were live S-type bacteria. So these R-type bacteria have been transformed, these R-type bacteria have been transformed into S-type by taking some substance from the filtrate. But here, as the filtrate was treated with DNAs, so in this flask, no DNA was present. And in the final flask, no live S-type bacteria were present. That means they could not take DNA from the culture which was present here and here. So in this experiment, no DNA was present, so no S-type live bacteria were found in this flask. So Avery, MacLeod, and McCarthy further showed that the purified transforming substance precipitated at about the same rate as purified DNA. It absorbed ultraviolet light at the same wavelength as does DNA. So obviously the transforming substance was DNA. These findings provided compelling evidence that transforming principle and obviously the genetic information resides in DNA and not in proteins or RNA.